So actually, I started the recording for the session too.
Okay, thank you very much. I will go ahead and share my screen here. So welcome back. Not that you went very far in the last like 30 seconds, but now we're in session 2, and we're going to do a first dive into PhysiCell, kind of showing a little bit of a recap of how to work with the sample projects or with the project you might have gotten from a collaborator. And then that will start kind of laying the initial steps to move us towards more advanced modeling workflows. Um, I'm going to be switching back and forth between several windows, so I'm not going to full screen my slides. Uh, if you're following along and trying to do this live, what I suggest is actually to have 2 command windows open, or shells, depending upon your operating system, just in your main PhysiCell directory. And just to kind of show how I've installed things, I installed PhysiCell, just unzipped the folder into my main user directory. And then I've also unzipped PhysiCell Model Builder from Randy Highland without the name of the version at the end, and also my user directory, so that I can kind of have an easy way to get in and out of these paths. So if you want to follow along, I just suggest having 2 shell windows open at the same time. And of course, you can download these slides and then follow along and kind of try it again later. And all the codes for the session will be available online in the GitHub repository. So the goals today is first kind of give a refresher from some of the materials from sessions 0 and 1 about sample and template projects and the overall structure of a project. We will briefly introduce cells, phenotype, and cell definitions. And then we'll learn about what the 3 major, general modeling workflows are for building models in PhysiCell: the basic, intermediate, and full modeling workflows. Basic we'll be doing now, intermediate in John's session in the afternoon, and that will really pave the way for the full modeling workflow starting tomorrow morning. And then we'll learn to just populate, build, and run basic models with a basic workflow, and we'll look a little bit at visualizing data in a Jupyter notebook. So some of the key background, just kind of this a little bit more of a refresher, is that PhysiCell comes pre-bundled with projects that are bundled with every download. And there are a variety of makefile rules to help you access those sample projects. Make list project gets a list of all the bundled projects. Make will compile whatever project you want to do. Oops, I should have added one more rule here. Make project name. So make with a project name, or this is called the sample name, will populate a sample project. So I will update these slides when we're done here. Make will compile that project. And then of course you run it with whatever executable is generated by that make file. You can run make cleanup to clean up the data in your output directory to get ready to do another run. And then make reset will clear out the project, the sample project, back to a blank slate, so that make list projects works again and you can switch to another sample project. Out of all the projects that are bundled with PhysiCell, the one that you probably want the most is a template project, which is the starting place for 2D and 3D projects. Throughout the workshop, we are primarily going to work in 2D modeling just because we want to be able to run examples very, very quickly for the sake of pedagogy. But the changes to a full 3D model are actually pretty straightforward. And maybe we ought to add maybe one asynchronous session on that. If you look into your PhysiCell directory structure, you'll see a variety of directories. Probably the ones that are most important to know are the config directory, which is where you store your XML configuration files. Custom modules is where you're going to be putting your custom code for your modeling project. And output is where your data are stored when you run a simulation. Of course, that can also be changed. And so remember that in PhysiCell, it has the cells really, as an agent-based modeling platform, are the key entity. And each one of these cells will keep track of what type of cell it is, its unique integer ID, its position, its velocity, its state, and in particular its phenotype, which includes methods and data to operate on that cell phenotype, and then custom data. And cells will have their own built-in techniques for division, death, changing their type, growing, shrinking, accessing and sampling the microenvironment, finding nearby cells, ingesting cells, and other behaviors that are regulated more directly by phenotype. Digging into the cell class, there are a few C++ bits that are good to know. that every cell has a string called basically its type name. So that's inherited, that's basically copied over from the corresponding cell definition. So if a cell is a fibroblast, it would have a string that says fibroblast. It will also have an integer type that's going to, where every cell definition has a unique integer identifier. And so that's an easy way for, the string types are for humans to look at what a cell is. The integer types are for algorithms to more easily compare and say, is it the same type or a different type? Because integer operations are way faster than strings. And then you have an integer ID, that's your unique cell identifier. You do store the cell's position and velocity as vectors. Just a word of note, you should not be directly writing the cell position, but rather use the update velocity and assign position functions, just because there are a lot of data structures under the hood that keep track of where a cell is in the environment. And so if you change the cell position without changing the underlying data structures, you're going to run into trouble in your simulation. And so that's why we provide other interfaces. But it is good to be able to sample cell's position and read it out. So we have made that accessible for now. Cell state will store things like size, pressure, and cells in contact. We'll get more into that in the other sessions. Phenotype is where you kind of group both the key behavioral properties and state of the cell. And that will be the topic of sessions 3 and 4. Custom data or custom scalar and vector data that you'll use in the course of writing custom functions. And then functions are where you write your actual cell tailored functions for your modeling. And that's going to be the big topic of tomorrow in session 7, I believe, or 5, I think, 7. So a little bit more about cell state. And this will be the topic of sessions 9 and 10. We start talking about cell contact. But every cell in state will store a vector of memory addresses of attached cells. And these are cells that you yourself manage. Actually, you can say format attachment, or detached cells. This is something that's under your control as a modeler for your specific models. Then we also have a vector of neighbors, and these are all the cells that the interaction potentials have identified as being close enough to interact. And so this is quite handy for your use later. Orientation is a vector used to determine the cell's division plane. So let me correct this just a little bit here. And so we can talk more about that later on as well. And then there's something called a simple pressure that's used for mechanobiologic feedback based upon the potential functions. That'll be a topic of later sessions. And then the cell phenotype is what's kind of hierarchically organizing the key behavioral elements of the cell, kind of those core activities we talked about in the earlier introduction talks. Cycle, depth, volume, geometry, which will kind of come up with an equivalent radius and surface area based upon the volume, mechanics, motility, secretion, which includes both outgoing secretion and incoming secretion, like net secretion, and for interfacing with the diffusion code. And then molecular is where we store anything that we've internalized in the cells, and intracellular is where you'll put intracellular models. And so putting all this together, really what we want to encourage is a way of thinking of phenotype-centric programming, thinking that the core methods, the core cell behaviors are already implemented. Cycling, depth, adhesion, repulsion, motility, secretion, uptake. All of these are already built into the cells in a way that handles interaction with all the underlying data structures to keep things running, happily inside of a simulation, which means that you don't need to write those core raw processes on the cell agents. And what you can focus on instead is saying, how often do I trigger these processes? How fast do I go through the cell cycle? What's my probability of dying? How strong is adhesion and repulsion? What direction is motility? Do I need to change direction? Do I change my secretion or uptake rates? And those are the kinds of things that you want to change as an agent-based modeler, rather than saying, I want to change the cell volume by 3 cubic microns, or I want to change the position by a little bit to the left. You want to kind of change the parameters and let these things kind of handle themselves. And that's phenotype-centric programming. We talk a little bit about this a little bit more in session 5, but there are cell definitions in PhysiCell, which basically is like a template for creating a cell. It will have the name, the type, as well as default values for all the parameter values in the cell phenotype. And so this will be a great way to kind of set up all the properties of your cell. So you say, I want to make a fibroblast. It copies over the default phenotype and it's already ready to go. You don't have to set a bunch of parameters for every cell you create. So let's talk a little bit about modeling workflows and then kind of jump right into the meat of it here. Before I go, are there any questions before I move on to workflows, and then we'll start doing some of the basic modeling? Okay, and I'm not seeing anything in the chat window, so let's go ahead and keep going. Thanks, John. So in PhysiCell, we like to think of it as there being basically 3 levels of modeling. The basic modeling workflow is something you actually have already seen in session 1 with the sample projects. The idea is that you would build an existing project, change the parameter values, run it, and visualize it. And so this could be either a sample project built in with PhysiCell, or it might be some kind of a code that you've inherited from somebody else, say from a team member. So this is what we'll work on today as a way to kind of get our feet wet with working with PhysiCell and prepare the way for an intermediate modeling. And that is where you're going to take use a graphical model editor that was introduced by Randy just a few days ago, developed all spring semester and then formally introduced last week. And this is where you can actually graphically set up the microenvironment, the domain, all the diffusing substrates in the microenvironment. You can set up all the cell definitions and the default phenotypes and save that to a file. And that's all ready to go as a basic modeling project. And so it's a really great way to get going. And then the full modeling workflow, which I'll introduce tomorrow, will take that intermediate workflow and then add on custom C++ functions to model your cell hypotheses. So you do a lot of the grunt work in a GUI to set up the environment and the cell definitions, and then you kind of work on C++ functions to model cell phenotype rules. And so that's where we'll be. So let's move into the basic modeling workflow. So the idea here is basic modeling workflows. Again, it's suitable really for taking a built-in project or a project from collaborators, and then just saying, you know, I want to explore this model. I want to change the parameters, run it, and see what it does. And so the key steps in this modeling workflow is to first, you know, populate one of the projects and compile it or build it, edit any of the settings in XML, run your model, and then view the results. And so that's what we're going to focus on for the rest of the session this morning. And so remember, we can use that rule, make list projects, to get a list of projects. And then for today, for our session 2, we are going to build the heterogeneity sample and work with that. And so we'll make list projects, make heterogeneity sample to put all the source files where they belong, and then type make. So I'm going to go to my command window over here. I'm already in my PhysiCell directory. So type make list projects. And you can see that lists all the sample projects, and then also the intermediate projects for intracellular models that were released in the newest version of PhysiCell. So I'm going to type make heterogeneity sample, and that will populate and put all the source files where they belong for the heterogeneity project. And now I'm going to type make. Here's a little nice trick. If you do a -J and say 4, it can do 4-threaded compiling. So basically it's running 4 threads for your compiler. So it compiled whatever files were changed and stitched them together into an executable. In Windows, those executables will end in .exe. In Linux, they are not. So you can see that I've actually run a few sample projects in this directory. And so here, if you're in Linux, you're going to type a dot, or in Mac OS, you type /heterogeneity, and that would execute this project. If you're in Windows, there's no // you type heterogeneity.exe to run it. Okay, so I think I saw the chat window bloop for just a second. Yes, these commands are the same for Linux, first of all. The -J, can't guarantee it will work on all machines, but it should work. But try just make by itself if it doesn't, and you can do single-threaded. If your make command says no rule to make target, then you need to go to your setup tutorials for PhysiCell, and we really can't stop for that here because that really needed to be done before the project started. But if you're in your directory and type in your PhysiCell directory, there is a make file there, and there's a make all rule, so there really should be a target. But we can try to troubleshoot that in the Slack workspace later this afternoon if you're still having troubles. Okay, so hopefully this works. The -J is only for make. for a multi-threaded making. The -J4 doesn't work for anything else. So you know what? Forget I ever said it. Just type make. Okay, so let's go ahead and get back to the session. Thank you. So now what we're going to do is we're going to go and edit the settings for PhysiCell. And what we want to do is we want to change the settings from the defaults just to make it a bit faster so we can kind of get some results very, very quickly and then spend more time on visualization for the rest of the session. And we will take a pause again in a few moments to continue answering questions. So kind of going into my PhysiCell directory here, I'm just going to go and enter it here. So here I'm in my home directory. Of course, use it however you want to for your own operating system of choice. But if I go into the config directory, for this project, I have a settings file called PhysiCell settings. So I'm just going to open this in my code editor of choice. So for me, that's just Notepad++, because I'm kind of an old school person here. Visual Studio Code is another great code editor. And I'm going to scroll through and start changing the settings. So I'm going to kind of put this on the left and my PowerPoint window on the other side. Oops, right and left. Okay. And let's see here. Let's change the domain to be -400, 400. So we're basically going to make a much smaller domain here. So I'm going to go here to Xmin and Xmax and change the domain size. You'll be building a lot of models in the GUI later on. But it's good to know the structure of the XML so you can make quick tweaks to a simulation rather than having to go all the way back into the GUI, because that's still kind of a work in progress. So we're going to change the domain size to -400 to this smaller domain. I'm going to keep scrolling through, and let's take a pause just a second on editing the file, and just note that in the settings file, the configuration is broken down to kind of major blocks here. We have domain. We have overall, which is where we set things like how long to simulate. We have some OpenMP settings in parallel, save settings of how often to save your data and where to save it, some other legacy options here, microenvironment setup, the cell definitions, some initial conditions. So I think that's actually a new thing here. So let's put that right after cell definitions. Oh, we should probably reorder this. So initial conditions really gives you some opportunities to change how you start your file. That is something that was introduced more recently in PhysiCell. And then user parameters. So let's go and change this order here just so that it is in the same order as the XML files, just to make this a little bit easier to navigate. Okay. So again, we'll update the PDF here of slides when we're all done. And then, so kind of going into these different blocks, let's go into the domain. And as we started earlier, we're going to change the domain size to be -400, 400 by -400, 400. We're going to leave the Z settings alone as basically half a delta Z down, half a delta Z up, remembering that a 2D simulation in PhysiCell is actually a 1 voxel thick 3D simulation. So we do need to have DZ over here. And then we set this flag use 2D to true to make sure they're going to run a 2D simulation. Let's continue editing some of these settings here. I'm going to go down to the user parameter settings and change some of the settings for our simulation. I want to add a little bit more heterogeneity to the sample. So I'm going to change the oncoprotein standard deviation from 0.25 to 3. So we have a lot more variability in the oncoprotein. And I'm going to allow we tend to cap the values of it in this particular initialization. So I'll set the maximum oncoprotein to 3 standard deviations plus the mean, or 10. And those are the 2 main things to change in user parameters. And then I'm going to go into save settings. Oops, since I'm not using this, turn word wrap off here. I'm going to go now into the save settings, and this is a little bit important. I want to change the maximum simulation time. I guess this is in the overall settings, sorry about that. The maximum simulation time, I want to change this down to 2160, so like 1.5 days. And I want to save data a little bit less frequently. I'm going to save data every 360 minutes and leave everything else alone. And let's see here. And I think that's it. We've done all the changes we're going to make in the settings. Let me take a quick pause here and see if there are any questions we can answer quickly. And it looks like Randy is actually answering a lot of them in line right now, which is just absolutely wonderful.
Hey, Paul.
Yeah, please.
I just wanted to, so yeah, okay, great. Randy just addressed Nikki's question. And then other than that question, I think we're all up to date on questions in chat.
Okay. Are there any questions I should answer right now, John?
I think we're set.
I think we're set. Great. I do want to point out that PhysiCell basically works natively in microns for space and minutes for time. And so if you change time units basically you need to use consistent units throughout PhysiCell. So you can't use centimeters here, minutes there, meters here, microns there. Really everything needs to be in microns and minutes because PhysiCell does not internally convert units. So please do be aware of that in the parameters. So it's best to just think in terms of microns and minutes if you can. And I will go ahead and leave this be right now. Now, if you can't run this in real time, that's okay, right? We're kind of going fast because we want to cover a lot of ground. But all this material will be available for you to run through it at a bit slower of a pace later, but I really wanted to have all this together in one place so you can kind of really see it and have a video to refer to when you're done. But definitely give lots of opportunity for questions as we go through and then particularly at the end. I guess, John, as moderator, if there is like a showstopper question, please kind of call it out while we're going, and then we'll make sure that we hit that showstopper question. Otherwise, let's go ahead and get to running the simulation now. So we've changed all the setting files in our editor of choice, changing like the simulation time, the domain size, and some of the scientific parameters. We're going to make sure that we save that file. It looks like we already have. And we're going to go back to our command window now. And I'm just going to type heterogeneity to run this file. And it's going to run this model. It's going to parse that configuration file and grab all the settings. So notice that a lot of stuff gets popped up to screen over here as it goes. You'll see your simulation domain size popping up here and with the number of voxels, a listing of all the diffusible substrates in your environment get listed, and a listing of all the different cell types in our simulation. And then periodically, whenever it does a full save, it'll spit out some information to screen saying, how many cells do I have in my simulation right now? How far am I towards my maximum simulation time of 2160? And I chose these particular parameters because it should take like 1 or 2 minutes for this to run. While it's running, let's go ahead and see some of the output. So let's see here, go back into PhysiCell. I'm going to get out of my configuration directory, and I'm going to go into output. And so you can see, right, it's generating a variety of SVG files. These are the simulation snapshots. It's a fast, scalable vector graphic. So it's plotting the simulation as it goes. Tomorrow, we'll have some reference material about how you can change the coloring of these cells to suit your project. But you can see these cells are getting colored, printing nice little spheres. And this is basically plotting the z=0 cross-section of our simulation. So if you have a 3D simulation, it will still plot a snapshot, but it's going to be plotting the z=0 cross-section of your simulation. So this is like a really, really fast way to get a sense of what's going on in your model before you do any post-processing. Other things that are being popped out, XML files, which give kind of like the metadata of your simulation. This is using a draft version of what's called multicell DS, a multicellular data standard. And if you open up one of those XML files, you'll see some metadata that could stand to be better populated, information about your microenvironment and your mesh. With an external map file where we store the actual mesh voxels and then the actual data of the simulation time. Then a list of cell populations using individual-based model representation, where we're going to save both some simplified outputs and then a more extended list of outputs, basically in a tabular format of all the cells. And then the .mat files are where we store the actual data. And we'll talk a little bit about how to access that in Python and in Jupyter at the end of the session. So let's see here. We should be just about, nope, we just finished running. So we're going to get into visualization. And there are a few built-in makefile rules to make visualization pretty easy. Oh, the other thing we want to look at, by the way, is something called legend. And so this will spit out basically for all the cell definitions, it will run whatever your coloring rule is and make a cell of that type and pop it up into this legend. Now, this simulation does not have a very exciting legend because there's only one cell type, cancer cells. But other simulations like, say, COVID-19 might have 10 or 15 different cell types, and then this legend gets kind of interesting. Let me take a quick pause here. And John, are there any questions we should answer?
Hi, we've got one quick question. Genevieve asked, and I didn't quite catch when she was asking this question, so maybe as needed, maybe Genevieve can address this. She asked, when running on a cluster, will this be piped to standard out? So
that's a great question. Yes, it should pipe to standard out. Now, whether you have that standard out attached to anything or not is another matter. It may or may not actually capture. But I think you probably use like basically the alligator marks, the greater than signs, and I think you can actually specify that to go into a text file instead, for example. But yeah, I guess those would be going to standard out. Whether or not you actually see them is another matter, right? Depending upon your cluster setup, but you can probably pipe them into a text file.
And she has another open question.
Yeah.
Is it possible to input a specific mesh to create a specific topology? I thought you might want to address that.
That's a great question. Well, the domains are Cartesian right now because the underlying diffusion solver uses a locally one-dimensional method, which has been optimized for a Cartesian mesh. It doesn't have to be, equal dimensions in all three directions. It doesn't even have to be equal dx, dy, dz. That can be specified. If you want to simulate something that's a little more complex, it's something we used to do with like DCIS. There are ways to basically not fake, but more or less to approximate a more complex domain. And what you would do is basically you want to embed that into a larger square domain. And anything that's outside of your domain, you might want to set as what's called a Dirichlet node to set it to basically a constant value for Dirichlet conditions. It's like far field values. And that way the diffusion solver is going to do diffusion across the whole rectangular domain, but then overwrite the value with your Dirichlet settings, and then just do diffusion of parts that aren't diffusion, aren't Dirichlet nodes. That will approximate Dirichlet conditions on a regular domain. We don't have Neumann conditions on the regular domains yet, because that would take a little bit more work. That's something that would be a really fun project, for example. We also don't have, say triangular meshes and things like that, just because the underlying diffusion solver has been optimized for Cartesian. But if we wanted to couple to some other diffusion solver, say something like an FE Bio, then that would be a possibility, and it's a really, really good thought. Any other questions before we move on? Okay, so what we're going to do is now we're going to go and do some quick visualization. I'm going to type, and these do rely upon you having Image Magic installed. So if you don't, don't worry about it. Use your favorite, image routine of choice. But Image Magic has the benefit of being fast and cross-platform and available just about everywhere in the universe. If you type make JPEG, it's going to use Image Magic to do a batch conversion of all the SVG files into JPEG format with dimensions that are divisible by, I think, 4, so you can use them in an MPEG-4 movie if you want to. So we'll do a slight resize or trim as needed to make sure that everything has the right size. And this makefile rule does assume that you're in the output directory and not, say, output 1 or output 2 or some other directory. So please do be aware of that. But now you can see that there are a whole bunch of JPEGs that have been generated. If we go through another rule that's handy, you can make an animated GIF with make GIF. Although it turns out that actually, I think make movie is actually a little bit faster than make GIF. So here, this will use FFmpeg, which is built in with every download of Image Magic. So another good reason to get Image Magic is you get FFmpeg for free. And that took all those JPEGs we just made and turned it into a movie called out.mp4. So let's take a look and see what we did here. So we have our simulation that starts off as kind of a salt and pepper distribution of cells with different oncoprotein values. Yellow cells proliferate faster, blue cells less fast. And if I just kind of scroll through my simulation frames, or I can just play it here, you see that the yellow cells are preferentially dividing and kind of outcompeting the other cells. And then eventually this thing gets a little bit too big and oxygen gradient grows, and you have these brown cells in necrotic here in the center. So there we have a basic simulation that ran in just a couple of minutes. We were able to change some of the parameter values in the basic modeling workflow and then visualize and run and visualize the results. And so what we want to do is spend the last 20 minutes or so looking in a Jupyter notebook to kind of get at the data that have been generated by this by the simulation. For most of our tutorial purposes throughout the rest of the week, we probably won't be working in Jupyter just because we don't really need to get into the nitty gritty of the model outputs. But I really want to make sure you know, first of all, that there is a code available for you to load the model data right into Jupyter. And certainly that could be the basis of other tools, for example, or could stand to be updated and improved. So we're going to load the model data into Python. So the first thing we need to do is go and get the Python loader. So I'm just going to copy this link here, and I'm going to go into the GitHub repository, into Python loader. And it actually turns out you only need one of these files. You need pymcds.py. So you can download the whole repository if you want, or you can just oops, that's not what I needed. I'm going to click on raw here, and then just save this thing as a .py file. And I'm going to just save it right in my PhysiCell directory. So I'm going to go into my home directory, go into PhysiCell, and notice that my browser wants to save it as a text file. So you're going to have to click on all files and get rid of that .txt at the end and just save it as pymcds. It looks like I've already done this, so I'm just going to overwrite it. So if we save this file here, or download the whole thing and unzip it and grab this file, pop this into your PhysiCell directory, and we will have what we need to download and look at basic simulation data. And then the next thing we want to do is let's go ahead and start either interactive Python or start a Jupyter notebook. What we're going to do actually is we're going to just start a Jupyter notebook. If you look at the there's a hyperlink in the slides here today, and also it's in the code directory for the session, but you can download this Jupyter notebook that we'll be using here today in just a moment. And I suggest just put that Jupyter notebook right there in your root PhysiCell directory so we can access the data. So I'm going to go ahead and go into Jupyter rather than into interactive Python. And you can start, if you added Anaconda to your path in Windows or in Mac OS or in Linux, you should be able to run this directly from the command line. So I'm just going to type Jupyter notebook to start a Jupyter notebook and then open directly this particular notebook. So we see now we have this notebook running from the root directory in PhysiCell. And we're going to kind of run through these in different steps. Now, all this stuff is documented in my PowerPoint slides, but I think I'm going to spend the rest of the session here just in the Jupyter notebook, just because all the commands are already handy there. And what we'll do is we'll kind of go through these blocks one at a time and explain what's going on here. So first, in like any Python session here, we need to load, modules and dependencies. So we're going to import, the pymcds method from this Python file we just had. Import, like the standard stuff you always import, NumPy and matplotlib in this first directory. So you just kind of go into that first block here if you haven't used Jupyter before. But we're going to hit Shift and Enter to execute that block, and it's going to load those modules into memory. And now I'm going to uncomment this and just type pymcds for that main method, hit dot and hit tab. And you see how it's going to give you a drop down of some of the various methods and things that are available in this pymcds module. So that's just kind of a handy way to quickly learn what's available in pymcds. Let's go ahead and skip that tab. And then let's start talking a little bit more about loading data. So if you're in your PhysiCell directory, the main command to load data is pymcds. The MCDS stands for Multicellular Data Standard, which is kind of our draft data format for model outputs. And so you type the name of the XML file. So, look in our output directory, let's sort it by type. And we have a whole bunch of things in this directory, right? I suppose what I could do here is I could do we can see all of our XML files over here. And so I have, we saved, the initial time and then 6 more simulation times. So I'm going to read the initial simulation time, and then say that this should be looking in the output directory. So you kind of say the file name and where to load. So if you are in the output directory, then that would just be /, right? Because you're in that particular directory. But here we're in output. So, and I'm going to save the results to this MCDS, just as a data structure. So MCDS is just a data structure. And actually, I'm going to do this. I'm going to go ahead and split the cell. If I can. Let's see, I'm trying to remember here. Edit, split cell. There we go. Okay, so what I want to do is kind of actually browse and show you what is here in this. So let's try this again. So edit, split cell. There we go. So MCDS is this. We've loaded the data. Thanks, Python. I'm just going to type MCDS into tab and kind of see what's in this thing now. But you should see, oh, that's not quite what I wanted. So let's just keep going with the notebook. I'm going to kind of show you the data and the keys and what's in here. And so, for example, you can call mcds.getTime to say what simulation time was saved in that data set. You can get a list of all the cell variables, which can be handy. You can get a list of all the cell substrates. In this case, it was oxygen. And also, we can look at the data and get more of the keys. And this is really the important thing, really. The MCDS has metadata, has mesh information, has continuum variables, things like oxygen, right? And then a list of discrete cells. Let's take a quick pause and look at the questions. So some ask questions about Dirichlet conditions. Yes, we definitely support mixed boundary conditions where you could have different Dirichlet conditions on different boundaries, or you could even have, Dirichlet conditions on some boundaries, not on other boundaries. We don't, in the XML, have a capability of saying that there's a Dirichlet condition that varies along the boundary, but you can write that yourself with Dirichlet nodes. And so maybe that's something we can get into later, but basically you can specify the value at any voxel at any point in space and time as a Dirichlet node and set a specific value. You can even vary that in time if you want to. So you can use that, for example, to approximate far field conditions. And it doesn't have to be on the edges, but if you want very fine-grained control Dirichlet conditions, then you want to go in and start using what are called Dirichlet nodes. So that is possible in PhysiCell, actually with the underlying BioFEM with a little bit of more advanced use. John, are there any other questions in the chat window I should be looking at?
I don't think so. I'll just field this to you briefly. I answered, but Rishma asked about if there are different numerical, what specific numerical methods are used for solving systems, and if there are ways to change it. I don't know if you'd want to address that.
So I can address that a little bit here. The underlying diffusion solver is using the method paper in BioFEM, which uses a locally one-dimensional technique with operator splitting and a finite volume method, which has been basically using center differences in time and implicit solver in space in each X strip or Y strip or Z strip, which gives you a tridiagonal linear system, which then is numerically stable, second order accurate in space, first order accurate in time. And you can solve that, and we solve that directly with the Thomas algorithm, and then parallelize with OpenMP. So that's all there. We basically chose that as a nice compromise between simplicity and scalability and performance and accuracy. Within PhysiCell itself, the velocity and position updates are using, as John pointed out, the second order Adams-Bashforth method, which allows you to basically say, use the current velocity and the previous velocity to get a second order accurate in space update on the cell positions. And that's possible just because you can cache the velocities and get something out of it and use fixed step sizes. Right now, that is the cooked-in technique. It is possible to build other techniques, but it would take some work. And so you have to make trade-offs between in a completely open architecture where you don't know what numerics everyone is using, that can vary from simulation to simulation, or just try to get one set of numerics going well and will support it. And so we've opted towards that to say we have one thing that we know what's going on. We try to tune it as well as we can to this scenario, but there's certainly going to be some room for improvement. and I think there will also be some room to replace some of the numerics with others. Now, some codes, particularly where they're highly nonlinear, you don't necessarily have all the information you need to do to necessarily look forward in time and do fully implicit methods. And we haven't really gone to the trouble often to do multi-step solvers, but we've picked fairly small delta t's and tested for convergence to get methods that are first order accurate in time with forward Euler stepping. Some of the solvers are backwards Euler just because they were reasonably easy to do. And if you look in the PhysiCell method paper, you'll find a lot more information on that. So those are really, really good and deep questions. So thank you, John, for posting the link to the Jupyter notebook. And yeah, this is also linked from within the slides itself. So if you go into the slides, let's see here. Go back just a little bit. The link to the Jupyter notebook is first on this Getting Started on IPython page. For what it's worth, all the commands you could directly paste into a Jupyter notebook, so you don't necessarily have to have that notebook to go, but it's good to download it if you can. Okay, so we have loaded a data set and just kind of seen the overall data structure of what's in this MCDS data structure. What we really want to do, these code sections are not very descriptive. Let's go back to the slides and then execute in the notebook. So we downloaded the single time. We printed what's in there. We looked at the keys of what's available. And this in the Jupyter notebook is code section 2. What we want to do now is start looking at some of the cell data. And so for this heterogeneity example, what we want to do is first of all, get the mean value of that oncoprotein. Remember, that's the parameter that we allowed every cell to have its own individual value of it. So there's going to be some kind of a distribution. So we're just going to, first of all, let's go into the notebook. We'll get the mean value of that oncoprotein. And then we're going to start doing some plotting. So I did just a little bit of changing, some of the defaults in Matplotlib for the plot sizes. And we're going to plot a histogram of those oncoprotein values. And this is all in block 3 in the Jupyter notebook. So I'm going to hit Shift Enter first to get the mean of the oncoprotein. This is at the initial time. And remember that we said the mean was supposed to be 1. But this is basically applying a normal distribution, but cropping it, saying that the values can't go below 0 and can't go below 10. That added a rightward bias, which basically shifted the mean upward. And so that's why you're seeing that there. If you're really looking at the parameter value said we wanted a mean of 1, standard deviation of 3, but because a whole bunch of that bell curve was truncated because we don't want negative values, that shifted the mean to the right. And we probably could have picked a better distribution. And now I'm going to import Matplotlib and set some of the parameters and then make a histogram of those oncoprotein values. So now you can see, this is basically looking like that kind of that truncated histogram, just because we don't allow negative values to that parameter and we truncated at 10. So we can see that, there are a whole lot of cells with a low oncoprotein value and then kind of a tail off into the higher values here at the initial simulation time. And that's kind of shown where you see that histogram. Remember that first plot was mostly blue with a few specks of brighter yellow with a higher oncoprotein value. And so that's really reflected in this histogram. So now let's plot all the cells. So we're going to go and mcds.data has a key called discrete cells. And that discrete cells itself is a data structure that has a whole bunch of properties of the cells. So we're going to get the X position, the Y position, and the oncoprotein value and just store them in these variables. We'll do a scatter plot coloring the cells by the oncoprotein value. And then maybe we'll at least make the axis square to do a better plot. And this is all in Jupyter Notebook section 4. So we'll run these 2 commands. We'll grab the values of the X positions, Y positions, and oncoprotein, and then we'll do a scatter plot and rescale the axis a little bit here. And notice that this is a really ugly plot, right? We're not even plotting squares as squares right now. So there's some things we could do to clean up the plotting here. And the cells, one weakness of Matplotlib is by default is plotting things by like pixel sizes instead of by the cell's actual physical sizes. So that takes some work. And Randy has some fixes for that if you want to do more advanced plotting. What we're going to do is we're going to start improving the scatter plot a little bit. And so we're going to change the size of the circles. We're going to make sure that squares are squares and circles are circles using the image command on the axis. And we'll add a color bar and nice labels and titles and things. So this is all in Jupyter section 5. So I'm going to clear my plot, although it's not necessary in the notebook. Redo my scatter plot, change my axes a bit, put on pretty labeling, and now we have a much better plot. And by the way, we could have done, there's another command that I'll show you later on in the notebook where you can change the default window size in the Jupyter notebook so that you don't have these really, really tiny plots. So now what we want to do is let's load a different simulation time. So now we'll load that final simulation time in that 6th output, and then we'll go and do, we'll grab the simulation time, and we'll do a nice title and histogram of the oncoprotein and see how that's changed. This is code section 6 in the notebook. So Shift Enter, and now you see that the this histogram is quite a bit different, right? We have, still some cluster of the lower proliferating cells, but then we have a huge cluster of cells with a higher oncoprotein value. And here you're starting to see that selection process. The cells kind of in little regions have kind of grown clonally, and cells with one of the higher values of the oncoprotein that have been more aggressive, faster proliferating, have come to dominate the simulation. So you can kind of see that selection process right here in the histogram. Now let's I want to show you some more advanced techniques that you can use, for example, to find the live and the dead cells. And so in multi-cell DS, cell cycling will have like a model code. And any model code above 100 denotes some kind of a death cycle, and anything below 100 will denote something that is a live cell. So that's an easy way to do some filtering, where you can say, we're going to grab the cycle and turn it into an array of integers, and then we're going to search for the indices where they're live, search for the indices where they're dead. Let's go into code section 7 in our notebook. And, oops, and basically we got it. What I'm going to do here is print live, for example. And here's my list of all my live cells. We can get, I suppose live dot size. Oh, I'm used to C now. I do like length of size or something. Sometimes it's hard for me to switch between Python and C because I'm just so used to C now. So there are 2897 live cells. And there are 124 dead cells at that simulation time. So that's one easy way to start getting some data. Let's look at instrument analysis. In section 8, what I want to do is start doing some plotting only with the live cells. So we'll get the count of live and dead cells. Let's get the mean value of the oncoprotein in the live cells only, and then we'll do a scatter plot of only the live cells. So we'll not plot the dead cells. And this is the next section in our Jupyter notebook is section 8. So you can see we have 2897 live cells. The mean oncoprotein, oops, we should have maybe done a print around this. Maybe I should save that again. So we have the mean oncoprotein value has moved up, right, because of this selection process. And now we have this nice plot here that only shows the live cells colored by their oncoprotein values. Let's get to the end of the notebook, and then maybe we'll have some time for a few questions before we get to our break. So I want to show you just a little bit more data loading. And again, these are kind of printed here for your reference. So you can go back and use them as you look at your own projects. But let's do some, Python lets you do fancier searching. So for example, I can look for all cells that have cycle less than 100, so those would be live cells, and they have an oncoprotein value that exceeds 5. So you do this, and then you kind of flatten this into a nice flat integer index. And then that's a nice list of indices of what cells to plot. So then you do a scatter plot of the x positions of those cells, y positions of those cells, oncoprotein values of those cells for the coloring, and I'm just going to do slightly smaller dots now. I just want to point out that the size that you plot is really going to vary based upon the resolution of your desktop, of your web browser, the domain size and everything. So you may have to play with this s equals parameter to get a good appropriate size. Let's go ahead and execute that here. Oops, it looks like this command didn't necessarily make it into my Jupyter notebook. And so what I'm going to do is going to copy this all over here. Paste, and I'm going to do a split cell. And let's maybe, so here's our plot. And I want to call this something like code section, I guess 8A, because I need to do something here. So I really apologize that it didn't get copied into the notebook. So in the past, I've always run this just directly. Let's see, I think I hit escape, and that turns into markdown. And so we have this 8A now. So I'm going to go and copy this, paste this over here. We'll call it 8A. And so this allows you to do fancier searching to plot only certain cells. So you can imagine doing very complex search criteria if you want to in your Python, and really attack at different cell populations. And so we have that plotted over here. So let's do a little bit more. Let's plot the oxygen. So you can plot substrate values. So I'm just going to go here and just do a standard contour plot. So we can get the names of the substrates, and then we can kind of, let's go ahead and split the cell here perhaps, just to kind of show you, that you might want to query to say, who am I, get my substrate names. Good Lord. Not helpful without a print. In an interactive Python, that would have printed. Okay, so we have oxygen as our kind of our main substrate name. And so we're going to choose one of those variable substrate names and do to go and get the concentration, and then create a mesh of X and Y, and then do a contour plot with those oxygen values through the z=0 slice. So we have now kind of a nice oxygen contour plot here for the final simulation time. And what we can do then is we can start combining that to plot the cells on top of the oxygen values. And so this is where it starts getting you more complex visualizations. So I'm just going to, the commands here, I'm going to set my circle size. I'm going to get the substrate names just like before, get the oxygen. So basically redoing this work, but kind of combining it now with the scatter plot. And I'm going to say, let's plot only the live cells and the scatter plot on top. So now we've put everything together and we have this nice, admittedly very tiny scatter plot of cells. So we have just a couple minutes left, but I want to kind of show you that you can do time series analysis by basically looping this. You can say, what's my lax index count, and you can just load simulations one at a time, generate the file name with, like sprint type commands by putting basically string concatenation here. And you can generate your simulation times, and which will tell you what your file name should be, load the appropriate times, save your results to an array of numbers, and then you can plot things when you're done. And so here we're just doing kind of a quick time series analysis, and it's loading all those data points. And then we're going to plot what we found. So we're going to plot like the live and dead counts, and even do some polynomial fitting to it. So we got that. So we can plot like the live counts over time, the dead counts over time. We can definitely see this tumor is growing. You do a polynomial fit to the logarithm, which would kind of give you an estimate of the growth rate, right? Because it's like a log linear plot. So this would be your units in minutes inverse. We can also plot, how the mean oncoprotein value is changing over time. We can see the selection process in action, that the populations with the higher oncoprotein value, the more aggressive tumor cells are proliferating faster. And so they're dominating this simulation, and the mean value goes up. Just kind of a last thing, we could do a make cleanup and do a make reset to kind of clear out the simulation data. And if you want to work through on your own, I have another example here using biorobots where you can show how to plot things with different cell identities. And so let's go ahead and pause it here and take some questions. And and I will update both the Jupyter Notebook and the PowerPoint slides. I'm sorry, the PDF output of the PowerPoint slides by this afternoon, so you can run all these commands and get some of the bug fixes in the slides. So happy to take a pause here and take any questions.
Great. Paul, where is the, so Jacobo asks, that he cloned the repository, the Python loader repository, and downloaded the Jupyter notebook too. How do I get PyMCDS as a module so I can import it? And
okay, so I'm a bit of a Python caveman. So all I did was actually go into my repository for PyMCDS and physically found that pymcds.py file and copied it into my PhysiCell directory. So this is Python for cavemen like me. There are probably fancier things you can do to add it to a path, but I'm kind of admittedly a Python caveman and not that advanced. We will definitely post updated slides and notebook by this afternoon. So that's answering Genevieve's post. John is looking into things. Eowyn, hi. So you have a question, looks like, how does oxygen diffusion interact with the cells? Is it uptake only, or are there other space considerations? So that's a great question. So we, in this particular problem, are solving the reaction diffusion equation, where we have Dirichlet conditions for oxygen on the outer boundary, diffusion and a very small decay constant going in, and then the cells are also uptaking oxygen. And so since the cells have, a non-uniform spatial arrangement, that's going to lead to the formation of non-uniform gradients throughout the cell simulation. And so as the cells move, the gradients move around. Where there are more cells uptaking, you get sharper gradients, which kind of eventually will lead to hypoxic and necrotic zones. Hopefully that answers your question. It's a really good one. You can get more and more interesting the more interesting things you do with your cell types. Are there any other questions? Julio, is the output data interval somehow related to the time simulation step? So let's go, that's a great question. What I want to do is go back into the configuration file. If you go to the very beginning of this file, you'll see some save information. And here, this says how often we save the full data set. So we are telling this to save the full data every 360 minutes here in the setup file, and that we're going to generate SVG files every 60 simulated minutes. So that is independent of your simulation step size, but it probably should be a multiple of your simulation step sizes. You may notice somewhere in the overall settings, we set the diffusion step size as defaulted to 0.01 minutes. This is something we have arrived at after a whole lot of convergence and accuracy testing in our original method paper as a good default step to get you good relative accuracy and good efficiency on anything with diffusion coefficients on the order of the size of oxygen or smaller. So that kind of order of magnitude, which is kind of typical for most of our biological problems. The mechanics step size is defaulted to 0.1 minutes. So mechanics will go not every time, but every 10 diffusion steps will do a mechanics step. And that is actually just to keep up with the typical 1 micron per minute type of scale for velocities. And then the phenotype is every 6 minutes, just because birth and death and growth processes are happening on the order of hours. And looks like we have one more question for Eowyn, asking about the number of compacted cells. Right now, yeah, that's a great question. We do not right now have any kind of explicit modeling of how cell compaction would vary the permeability of the tissue and therefore the diffusion coefficient. In fact, right now, in the current code, the diffusion coefficient is spatially homogeneous throughout the domain. I think that'd be a wonderful place to do a possible upgrade in the future, though. In particular, I would love to allow the diffusion coefficient to be 0 in some places and directions, so they can start modeling Neumann conditions or zero flux conditions on much more interesting domains. And yeah, the command was from pymcds import pymcds once you have that file in your working directory. And Eowyn, those are great questions, and we'd be very happy to follow up with you further. And that is actually one possible topic for, and maybe a more extended hackathon project, is to take the basic BioFEM and take a look at that Thomas algorithm and modify it to do a spatially heterogeneous diffusion step, diffusion coefficient, which would allow you to do some really interesting domains, but at the cost of a lot of extra memory use. So it's a trade-off. I'd like to do it just because then I can model my long sought Pac-Man simulation of Pac-Man running around in a maze. And for that, I actually need Neumann conditions on a maze. So someday. So we're actually eating into the break right now. So let's go ahead and stop the recording, please, and stop the session. But we will meet back here in, I guess, at 12:30